I'm going to take over his bit here. My name is Lisa Purcell. I'm from VIR, but from the active side of things, I co-chair the active trace group with Matt Hall. And so these effector function seminars we've undertaken under the leadership of a number of really great scientific forces in the field of effector function. And so I'll go through some of those names in a bit, but just to sort of set things up, we are recording this presentation, and all of the transcripts and recordings from the seminars will actually be made publicly available for review. And I'm sure Trey's going to put the link in the chat if you're interested. Just for everyone on the call, the attendees will be muted during the presentation, and we could unmute during the Q&A, which will be at the end for both presentations. And I'll try to facilitate those questions with our speakers. You'll see that picture at the bottom of this slide. You could just go into the chat and the Q&A section to add in your questions. Go to the next slide. So Joe isn't with us yet. He might be joining in a little bit, but he does work quite closely with us across all of the active initiatives. He's from FNIH. And so we'll go through some intros for Galit and Gestur, although I'm sure they need no introduction in this field at all. And we do have both of them talking. I think Galit's going to go first on polyclonal swarms and systems serology, and then Gestur on IgG allotype polymorphisms, one of my favorite subjects. And then we'll have a Q&A at the end. All right. Oh, and just to set this up, I've gone through this a number of times with all the other seminars, but just in case there's someone on who hasn't attended, the objectives here are really to sort of set the stage for what we know and what we've discovered in the last few years within the COVID space, but to set the stage around vaccine-induced antibodies, infection-induced antibodies, as well as monoclonal antibodies, and how antibody effector function actually influences efficacy in each of those spaces. And so this workshop has been ongoing since I want to say October, November. We've had a number of great talks so far, and we've reviewed a number of different subjects in depth. And so really looking forward to the talks today, and that will add to our host of subjects here. On the next slide. So just to call out, there are two groups that are really diving into effector function. One is this translation, it's on the right, the translation of preclinical effector function to clinical COVID experience group, and Galit heads this up, and under her leadership has brought in a number of people who are listed here. And this was one of the initiatives that they really wanted to take on in terms of a seminar series to set the stage. And then on the left, we have another group which are tackling a really complex subject, but trying to figure out how we could standardize effector function assays and whether or not we can pull in some of the information that we've learned in the clinical space to understand how we could introduce some of these types of biomarkers in infectious disease as we move forward. And so, as I mentioned, two awesome speakers lined up today. On the 15th of February, however, we have two other speakers, Mike Diamond from WashU, looking at mouse models, and Robert Kimberly from the University of Alabama, looking at FcR and IgG allotype polymorphisms. So mark your calendars. If you go on the website, you can sign up for the invite there. It is from 2 to 3:30 p.m. Eastern time in 2 weeks. All right, and so on to our two speakers today. So first, I'd like to introduce Galit. Currently, she's actually the head vice president of immunology and research at Moderna. I'm happy to say she's joined us on the dark side in industry, although it isn't a dark side. But she did her Ph.D. in experimental medicine and HIV at my alma mater as well at McGill. And did her postdoc at Mass Gen. There's over 130 publications on SARS-CoV-2 alone. I'm not sure if you knew that or not, Galit, but I looked it up on PubMed today. But really has pushed the field of effector function in really new and innovative ways, like our systems biology approaches, which she's going to talk about today. And then next up, we've got Gestur, who is a professor, PI, and head of IG research at Sanguine Research at Utrecht. And did his postdoc at the National Institute for Public Health and Environment in the Netherlands, which actually I think he had a dual appointment to the University Medical Center in Utrecht as well. Looked at Neisseria and immune responses to meningococcal disease. That was really interesting. We're gonna have to talk about that sometime, Gestur. Ph.D. In immunology from Utrecht and a master's at the University of Iceland in antibody-mediated functions. And again, powerhouse in this field, over 150 publications on effector function IgG across a number of different pathogens. And so we're really, really excited to have you here to talk about the IgG allotypes as well, Gestur. So with that, I'm going to turn it over to Galit. Just so everyone knows, we have about 30 minutes for each presentation and then time at the end for questions.
Okay, thank you, Lisa, and thank you for being our fearless leader throughout this effort to try to collect this community around this concept of FC, effector function. So what I thought I would do is I actually put the title for like the original document on here. So hopefully that's not confusing to the audience, but the idea really is to try to give you all an idea of the way that we've been thinking about antibody effector function from the perspective of how antibodies work together as swarms. So really thinking about them as polyclonal effector molecules that collectively drive different immunological functions. And the way that we think about these polyclonal swarm dynamics of antibodies to understand how they interact with the immune system is really by using different types of systems biology tools. And so what I thought I would do for today, for the next 25 minutes or so, is I'm going to take you guys on just a crazy whirlwind adventure where we're going to look at just a few of the COVID studies that Lisa mentioned that we did over pandemic to begin to understand how antibodies might be involved in controlling or clearing SARS-CoV-2, and then potentially at the end to think about the converging conclusions that are giving us some clue about the mechanism of action of antibodies against SARS-CoV-2, really from the lens of this systems swarms approach. Okay, so where we started thinking about this idea of looking at swarm biology of antibodies really began with this idea that we wanted to disrupt the vaccine dogma. And this was, more than 18 years ago through my work in HIV vaccine-related biology, and really thinking about the fact that antibodies really do so much more than just simply bind and block infection. And from work from Jeff Ravitch and Stanley Plotkin and Dennis Burton and so many other legends in the field, we've learned over the last 3 decades or more that antibodies are just doing so much more than just this single potential function. And these include the ability of antibodies to recognize infected cells or the pathogens themselves. And once these antibodies bind, their purpose is to essentially bring in the immune system to recognize this infected or aberrant material. And then these antibodies provide very specific instructions to the immune system on how those antibodies or how those immune cells should destroy anything to which those antibodies are bound to. And the idea really is that, we have an exquisite system built into our, B cells that allow us to produce antibodies with all these different instructions that are then used by the immune system to essentially destroy these pathogens. And so a few years ago, we set out to build a high throughput assay system that would allow us to collect information about how antibodies following infection or following vaccination can essentially interact with any of these immune cells, and to link that to the biophysical features of these antibodies to understand really what it is at a biophysical level that leads to a particular function, and to understand which combination of these biophysical and functional properties are enriched in populations of patients who are doing better with one particular infection or another, and to use this information to potentially inspire vaccine design or monoclonal therapeutic activity. So to start thinking about how we can build assays, high throughput assays that could interrogate all the possible functions of antibodies that could include phagocytosis and cytotoxicity and complement deposition and antigen uptake, we started to think about really what are the properties of the humoral immune response that we want to capture to begin to put into a machine learning exercise. And so when you think about immunoglobulins, as we've heard throughout this string of this series of talks, is we, here we're just using an IgG as an example, but we think of them as having, two functional domains. They're obviously a single molecule, but we have the top domain, which is obviously variable and is involved in antigen recognition, and it tethers us to the surface that we're trying to attack. But we also do have the second functional domain, which is the constant domain, which of course is the domain that links the innate and the adaptive immune system, and is the domain that essentially drives or provides instructions to the system on how it should destroy anything to which those antibodies are bound to. And we know that during an immune response, we make many changes to the functional domain. It's not really constant. And in fact, those changes, a collection of two different kinds of changes, can lead to different kinds of instructions to the immune system. So we can change a subclass of antibodies, which we all know in humans, we have 4 subclasses that give us 4 typical functions potentially, or flavors of functionality that we can elicit from the immune system. But we also change the sugars that are nestled between the arms of the antibody. And these sugars come in additional with more variation. So up to 32, I think, structures that can be added or at least reliably measured on the back end of IgGs in humans. And so what we think is like that combination of the right subclass and glycosylation is really, these combinatorial states that might give us different kinds of functionality, right? That could explain why we have so many ways that these antibodies can trigger different types of immunological functions. And we think that that combination of the subclass with the glycan is really the barcode that the immune system uses to drive different kinds of instructions. So you can have 140 potential different instructions that could include like deposit complement, activate NK cell cytotoxicity, drive antigen uptake, as just a few examples of what those barcodes could be telling the innate immune system to do. But the swarm dynamics is what gets exciting, right? Because during an immune response, we never make a single antibody to our target of interest, but during immune responses following vaccination or infection, we make billions of antibodies, right, to our pathogen of interest. And those billions of antibodies attack the pathogen in different ways, at different epitopes, with different affinities, with different angles. And it's really the swarm dynamics, or the combination of these antibodies that form these complexes, these immune complexes around our pathogen, that collectively together provide all the barcode instructions that lead to an immunological outcome. And if we imagine that during an immune response, we can form different flavors of these immune swarms, right, by vaccinating maybe with one vaccine platform or one adjuvant or through a different route of immunization, then theoretically what we have in our immune system is the ability to get more than just simple barcodes, 140 barcodes, but we have these much more complex barcodes that can essentially drive fundamentally different types of immune activities. And so what we set out to do a few years ago is think about how can we really understand, well, what barcode, what 3D barcode is associated with a good clinical outcome versus a bad clinical outcome, and how can we use this to really accelerate our understanding of correlates of immunity to pathogens. So this is really what led to this idea of trying to create a string of analytical techniques or different assays that could essentially capture information about the biophysical structure or activity of the antibody, including their specificity, their affinity, their avidity, their isotype subclass levels, their glycosylation profiles, to think about how they work together in this swarm biology or swarm dynamics, to ask what immune cells these swarms then can trigger or activate, and then to couple all this information to machine learning tools to begin to decode or ask what is the pattern that's associated with a good clinical outcome versus a bad clinical outcome, so we can build hypotheses about how we think antibodies are actually responding or driving disease control. And so this is what gave rise to this tool that we began to develop more than 15 years ago with one of our previous speakers, Margie Ackerman, with a tool we call system serology, which really is a tool that we consider really helps us to define unexpected correlates of immunity. I think we've been surprised often when we've done this exercise across pathogens. And so what I'm going to talk to you about today is how we use this tool to essentially look for immune profiles that are associated with protection or control of SARS-CoV-2. And just to run you through this, really this tool was inspired to look at human populations, but has been adapted over the years to look at lots of animal models, also to look at monoclonal therapeutics, which even if they're produced in a single cell line, often can have quite interesting and diverse functions depending on how they target the antigen, with what angle, with what complexity. And so the tool has been really quite revelatory to understand the interaction of antibodies with different types of immune receptors as well as immune cells. So we take the source material, whether it's plasma or fluids or different animals or monoclonals, and we run them through biophysical profiling or functional profiling. On the biophysical side, we just measure what is the overall level of subclasses or isotypes to up to 500 different antigens of interest using Luminex technology. We also ask how these antibodies interact with different Fc receptors or Fc-like receptors. And we also pull out the antigen-specific antibodies, and we ask if they have unique Fc glycosylation profiles, which we know this sugar is essential for regulating the affinity of antibodies for different types of Fc receptors. In parallel, we've developed assays, high throughput assays that are all bead-based essentially, that allow us to ask whether our pathogen-specific antibodies can fix complement, activate neutrophils, macrophages, monocytes, and K cells, eosinophils, dendritic cells. We have assays to look at non-immune cells like epithelial or endothelial cells, to activate non-innate immune cells like T cells and B cells, to drive apoptosis, to drive enhancement, as well as to deposit antigen onto follicular dendritic cells. And we try to think about how we can adapt this tool as rapidly as possible for our pathogen of interest, really trying to take into consideration really the types of immune cells that these pathogens might interact with. And we capture lots of data. And just like with transcriptomic analyses, we integrate all the data, we normalize it, and then we start to look for patterns in the data. And so this is just an example of about 100 individuals that were profiled for their flu-specific response. Every line is a different individual. Every column is a different analyte. And we're just looking for patterns in the data where you can see that individuals are really segregating, with some individuals on top that are hotter than individuals on the bottom that are cooler. And so we can use this data to begin to look for patterns using different kinds of analytical tools. Okay, so I'm going to take you guys now using this kind of background of how we think about antibodies and effector functions, and really the role of antibodies in directing the immune system to fight a disease. I'm going to take you on just a couple little vignettes of stories that we collected over a pandemic. And Lisa, if I get close to time, just stop me, and I'll just take questions. But for SARS-CoV-2, we got really interested in trying to understand how antibodies might be playing into controlling SARS-CoV-2, really because of the data that came out over the pandemic about the fact that these new variants were appearing, each that was slightly better than the rest at evading the neutralizing antibody activity elicited by many of our emergency authorized at the time vaccines. And this escape from neutralization really sent the global community into this boosting frenzy. Really people got worried because we saw these waves of transmissions occurring all across the globe. But what was really important was the fact that even though we saw these increased numbers of transmission waves, we did not see a concomitant rise in the number of severe diseases or death reports. So what this told us is that the vaccines had induced some flavor of immunity that was still controlling disease in the individuals, although it was not sufficient to block transmission. So what this told us is that there was something else in that vaccine-induced immune response that might be important. And while T cells were, of course, implicated, in many of the studies that I had done collaboratively with Dan Baruch throughout the pandemic, we saw that T cells were a correlate, but mechanistically, when we depleted them, that didn't really lead to a loss of control of the virus. And it was really the antibodies that were somehow conferring some extra neutralizing activity that was somehow dampening and clearing the virus more rapidly. So we got excited about this. And we started saying, well, let's look at human populations and ask if there is some special flavor of antibodies that's associated with better control of severe disease and death. And if we can understand what that magical function of antibodies might be, maybe we can use this information to really advance our understanding of how vaccines and monoclonal therapeutics control the infection. So where the story begins is really with a study we did back in May early on in the pandemic in 2020, where we collected samples from individuals who were admitted to the intensive care unit at the time. And we followed these individuals over the first month of their ICU stay. And essentially in this particular study, the idea was, this is early on, there were no vaccines, there were no therapeutics. We were asking the question of whether there was a natural antibody response that evolved in these individuals that could essentially help us understand why some people ultimately passed away versus others who survived the disease. And the idea here was we did all the work completely blinded to the status of the patient, and then only after we were done the analysis were we unblinded by our clinical colleagues, Ed and Rochelle, about whether or not the individuals went on to pass away or survive. And so I'm just going to show you just a snippet of this data, just to give you an idea that back in 2020, we already kind of had clues about what might be important in the control of SARS-CoV-2, or at least in COVID-19 severe disease. So what I'm showing you first is just the spike and IgG levels. These are just titers of antibodies from this ICU patients. And here I'm showing you these these levels from the first day of ICU stay at day 0, and just over the first 2 weeks of that admitted period. And what we're looking at here are just IgG levels to spike, and in the yellow are the survivors, and in the red are the non-survivors. And hopefully what you can see here is already at the first day of admission, survivors had higher levels of IgG specific, spike-specific IgG1s compared to non-survivors. But it wasn't that the non-survivors didn't try to make antibodies. I mean, in fact, they were probably making more antibodies faster than the non-survivors, but they just seemed to make them too little and too late. So we said, okay, well, binding antibodies doesn't tell you how these antibodies might contribute to viral control. So what I'm going to show you now is just 6 of the functions that we measured for these antibodies. So on the right side, what we have here are the ability of spike-specific antibodies to drive monocyte phagocytosis, neutrophil phagocytosis, complement activation, NK degranulation, NK cytokine secretion, or neutralization. And we're going to start here at the bottom. And hopefully what you can see here is that the red and yellow lines are right on top of each other. So neutralizing antibody responses did not evolve at different kinetics, individuals who survived or didn't survive. So neutralization did not seem, at least in this cohort, to be a predictor of differences in survival, or didn't act as a correlate of immunity. And K cell recruiting antibodies also did not differentiate the groups one from each other, whether we looked at degranulation or secretion. But we did begin to see was a striking difference in the evolution kinetics of antibodies that were able to drive opsonophagocytic function through monocytes or neutrophils, or complement depositing antibodies that all evolved much earlier in the survivors, and often, reached much higher levels over the 2 weeks compared to the non-survivors. So already back in May of 2020, we started to note that there was this difference in opsonophagocytic functions that seemed to be enriched, or a strong predictor of survival of disease in the natural infection setting. So this was really interesting to us. And so at this point, we started thinking, well, let's look in animal studies. And we did a number of animal studies to try to understand exactly which functions of antibodies induced by vaccines could be associated with better control of the virus. And one of my favorite studies is a study that we did with Bali Palendran, who's a friend at Stanford. And Bali did a really fun study where he essentially took non-human primates and he immunized them with a SARS-CoV-2 receptor binding domain nanoparticle. And he gave that nanoparticle in the presence of 5 different adjuvants. And so he wanted to ask the question, are adjuvants performing differently from a protection perspective? And if so, are there particular immune profiles that could explain the difference in protection afforded by each of these adjuvants? So I'm going to just show you the isotype levels, because it's pretty interesting to see really how these adjuvants perform. And the way to read these graphs is we're profiling antibody responses after the first dose, day 21, or after the second dose of the vaccine, so day 21 and 42. And then the color or the top of the chart tells you which adjuvant was used. So oil and water, AS03, which is an emulsion from GSK, AS37 is a TLR7 agonist from GSK, CpG alum is a TLR9 agonist, and alum, of course, is a widely used, we believe, TLR4 agonist. And hopefully what you could, or actually it's not at all, it's a different agonist. But anyhow, what we could see here is that after the first dose, you could see IgM responses appearing to the RBD across most of the adjuvant vaccinated arms. After the second dose, though, we saw that these responses were augmented in many of the groups, but interestingly, AS03 here induced the highest level of IgM, so the RBD, compared to the other groups, suggesting that at least for IgMs, AS03 was the clear winner. For IgG1s, after the first dose, we see clear induction of IgGs to the RBD. And after the second dose, they all increase. Again, AS03 being the clear winner, the most homogeneous levels of IgGs induced. But interesting, when we look at the IgAs, while everyone induced responses after the first and the second dose, it was actually AS37, a TLR7 agonist, that induced the highest levels of IgAs across the group, suggesting that there is this biological difference in how these adjuvants are triggering different kinds of isotype specific responses. So that was a first very interesting observation for the study. We also looked at the functions of the antibodies. And so here I'm just showing you NK cell function, NK activating antibodies, neutrophil phagocytic antibodies, monocyte phagocytic antibodies, or complement depositing antibodies, all to the RBD. And what was interesting in this context, looking at the functions, is after the first dose of the vaccine, no matter what the adjuvant was, we saw a very low level of functionality. So it really took a second dose to boost those responses. But when you looked at the second dose, what you saw here was that that second dose, really when you had AS37 and CpG alum, you got the highest level of NK cell activating antibodies. It wasn't AS03 that had the highest IgG titers. For neutrophil phagocytosis, it was a different pattern now. Again, well, so first dose doesn't give you much. Second dose, then we started seeing that the AS03 was the big winner on inducing neutrophil phagocytic activity. We didn't see much difference in how the monocyte phagocytic activity was induced. And we did see with the complement depositing antibodies that there was a clear winner here with AS03 inducing the highest level of complement fixing activities. So this was really fascinating to us. And if you make a composite at peak immunogenicity of the responses induced by each of the adjuvants, where these radial plots show you the median level of each of these bioanalytes in the blood of the monkeys, hopefully what you can see is the patterns are super different, where oil and water is, kind of a contracted, not very strong response. AS03 shows this kind of fuller subclass and FcR response, but this variegated functionality. AS37 is a little bit more balanced, although kind of intermediate. CpG alum is pretty full, but biased towards more NK cells than opposite phagocytic functions. And alum, interestingly, again, is kind of this medium response, but again, tipped towards more NK compared to opposite phagocytosis. So this is exciting to us. We had all this variation. So we said, now let's put this all into a model and ask, well, now that we have these different functions, what predicts protection from infection? So Bali challenged the animals, and here I'm just showing you the viral loads in these animal groups. And so here you can see in the non-vaccinated group, we have all animals 4 of 4 became infected, oil and water 3 of 4 became infected, AS03 1 became infected, AS37 2 became infected, so on and so forth. So we saw this variation in the level of infection where some arms were clearly more protective than others. And we looked at viral loads in these animals once they were infected to see what their level of viral replication was compared to their neutralizing antibody activity. You saw this beautiful inverse correlation. Clearly, neutralizing antibodies are tightly linked to the level of viral replication. But what struck me and I thought was always so interesting is that there was this group in the middle that had essentially equal levels of neutralizing antibodies, but had wildly different levels of viral replication in their blood. So neutralization is correlated, but it's clearly not the whole story. So we said, okay, let's take neutralization. Let's take all the data we have. Let's put it into a model and let's ask why these guys have high viral load versus these guys have low viral loads, and ask what is really associated with protection or a complete shutdown of the virus following a challenge. So we took the animals, we put them into the model, and what you can see here is that each dot represents a different non-human primate that we analyzed. The multivariate profile included all of the antibody features that we had captured. And just using all this antibody data, we could clearly separate out the animals who were completely protected in the green from those that had some level of viral replication in their blood. When we asked, well, what features are driving this difference of essentially protection versus non-protection, what we saw was that there was, I'm sorry, the animation is doing something funny, but what was very clear was that there was 3 features alone that were essential for essentially predicting protection or non-protection. And these features were all enriched in animals who were fully protected here in the green. And they included S1 specific antibody binding to an Fc receptor called Fc gamma R3, the ability of antibodies to drive neutrophil phagocytosis, as well as IgG antibodies. And these were all enriched in the animals. And interestingly, we could unblind to see which adjuvants drove the best activity. And it was really this accumulation of AS37, alum, and AS03 immunized animals that were all found to the right, and they all had this profile that was highly associated with protection. The reason I got so excited about this profile, though, is it reminded me of the humans, the naturally infected humans, where we see this neutrophil phagocytic function again appearing. And Fc gamma R3 as well, which we know there are two kinds of Fc gamma R3 receptors in humans. There is the Fc gamma R3A receptor, which is abundant largely on NK cells, but other types of immune cells as well. And then Fc gamma R3B, which is a GPI anchored sister of 16B, but does not have a signaling domain and is largely restricted to neutrophils and some basophils and eosinophils. And the reason this 16B was so exciting is because we knew there was enrichment of neutrophil phagocytic activity. And also we knew that neutrophils are these unique cells that express at least 3 different Fc receptors. This Fc gamma R3, which we saw elevated binding, was a correlate of immunity in the monkeys, but it also expressed Fc alpha receptor, so the receptor that responds to IgA, which is also part of the correlate in the monkeys. So all the data really pointed to neutrophils as being the primary effectors of viral control in the non-human primate model. And that's keeping in mind that in that model, we had neutralization, but it was not picked as an actual predictor of control of the virus. So the last story I want to take you guys through is the story now with convalescent plasma, because here's like a completely different model where we started to see that there was some therapeutic benefit in some patient populations. But the reason convalescent plasma was protective in some people and not in others, or in one trial versus another, was not entirely clear. So what we thought we would do is we would try to use system serology again to just objectively profile antibodies in individuals who, from the plasma donation to the individuals who received the antibodies, to begin to understand if there was any particular antibody profile that was associated with better control of the virus. So this was a really fun study we did with Lizanne Porofsky, who's a friend at Albert Einstein, and Lizanne was very active in the convalescent plasma trials. She worked with Arturo Casadevall, who presented in our first talk of the session. And what Lizanne really came up with is this idea that we could essentially profile responses in the, can I do this one really quickly, Lisa, and then I'll stop? Okay, so we could look at the plasma responses in the plasma donation and try to see if we could predict how somebody was going to respond or not to therapy. So we did profiling on all the plasma donor plasma antibodies, and what you can see here is every one of these plots represents a different donor plasma that we profiled. And so you can see here small radial plots, here are big thick radial plots. All the features are shown down here. And you can see here that every plasma donor profile was different. So everyone had their own snowflake of antibody profiles in the donor plasma. And interestingly, when we look at the patients, already at day -1 before the therapy came in, they had these massive responses to SARS-CoV-2, all different flavors of antibodies. And you can see that they had more and bigger responses compared to what the donor convalescent plasma therapy actually had. So we asked the question, well, okay, does this CCP affect the patient profile, and can that give us any clues about what might matter for control of the virus? So what was interesting is that we just wanted to look at the kinetics of evolution. We knew from the Regeneron studies that Lisa was part of back in the day, that there was a difference in how monoclonal therapeutics affected the outcome of patients following monoclonal therapy, depending on what their antibody titers looked like before therapy. So if they had high titers, they were less responsive than if they had low titers. And so we wanted to just look at the overall kinetics of the antibody evolution in these treated patients. And you can see here, some people started high and they stayed high, others started low and then increased over time. And the convalescent plasma was administered somewhere here between day -1 and 1. And if you look at the responses in the people who received the plasma, you can see here that the responses clearly evolved over time. So no big surprise. But the question was, well, what does it look like in individuals who had high titers to start off with versus low titers to start off with respect to their response to the CCP? And hopefully what you can see here is whether they had high responses or low responses, you can see that the profiles didn't really look all that different, at least by eye, when you looked at the aggregate signature. But what changed in their profiles was really highly dependent on the convalescent plasma therapy antibody profiles before it went in. So if you look at the low antibody titer individuals and you ask what in the patients changed over time, well, we saw, this is just a way to look at essentially the correlation between the two profiles. As you can see that there was like a whole bunch of different correlations where yellow was positive, blue was negative. We're more interested in the negative, like how do we stop the evolution of certain types of immune profiles? And hopefully what you can see here is that in the people who had low IgG ones versus high IgG ones, the profiles are very different across the two groups. So they respond to CCP in different ways, which to us was already kind of an interesting phenomenon. So here you see a lot of blue on the right. You don't see that here in the low IgG ones. They're doing something very different in response to CCP. And the question is, well, what's changing? So we started noticing these strips along these low IgG ones of how they're responding to the convalescent plasma. And they're doing it in a different way than how the high IgG titer individuals are responding to the plasma therapy. So I want to ask, well, what is most different? What is the CCP really affecting? So if we do this is the last data slide, and then I'll sum up. We can ask what is really changing dynamically due to the administration of CCP. So we're asking what are the features that are really like pushed by CCP. So over time, if you look at these features, the ones on the top are positively correlated with, they basically increase following CCP. And these are the ones that decrease following CCP, or they drive a decrease in the evolution of inflammatory responses in the patients. And hopefully what you can see here is in the high individuals who start high and stay high, these individuals really benefited from nucleocapsid specific antibodies. So if the donor plasma had high levels of nucleocapsid IgG3s and IgA1s, these individuals progressed slower and did better therapeutically than individuals who had received donor plasma that did not have nucleocapsid antibodies. Conversely, in the people who had low spike-specific IgG1s to start, these individuals did not benefit so much from the nucleocapsid-specific antibodies, but they did benefit super strikingly from spike-specific neutrophil phagocytic antibodies, as well as some S2-specific responses that are probably cross-reactive. So this told us is unexpected correlates. In the low IgG ones, just like in the normal progressors and in the vaccinated monkeys, spike-specific neutrophil phagocytosis was a correlate of protection, essentially controlling the virus, getting rid of it, and getting rid of inflammation. Whereas in the people with high IgG1 spike-specific titers, they really benefited from nucleocapsid-specific antibodies. Okay, so I'm going to skip over this. Lisa will not let me do this part. And I'm going to leave you with this last kind of thought that opsonophagocytic antibodies that may not be neutralizing, but they might have neutralizing activity, really were associated with natural control and resolution of infection. We saw that adjuvants could control these effector functions and drive different types of antibody profiles. And in the non-human primates with challenge, neutrophil phagocytic activity through IgG, through Fc gamma R3 and IgA were clearly implicated in protection. And in the convalescent plasma, we were really struck again that the spike-specific neutrophil phagocytic signature came up again as being critically important for protection. But we also saw this really unexpected role for nucleocapsid antibodies in therapeutic benefit in patients who already had high levels of spike-specific antibodies. We think that that's happening by removing immune complexes from the lung that essentially will drive some clearance of inflammation and pathology at the site of infection and replication. So I'm going to leave you with this last idea here, right, is that antibodies on a single molecule level have this intrinsic capacity to interact with the immune system in different ways. And we're learning that subclass and glycosylation control that biology. But during an immune response, we're making these swarms of antibodies, these billions, if not trillions, if not gagillions of antibodies. They work together to drive those kinds of instructions in the immune system. And so thinking about how we can decode that in, in vitro high throughput systems that help us think about how we can make better therapeutics and better vaccines that can get us to that end goal faster, may help accelerate the path to protection against different infectious diseases. And the last thing I want to leave the audience with is just to think about the fact that, we're thinking about canonical immune cells and canonical receptors, but there are so many other types of immunological mechanisms that we haven't even tapped into because we're only thinking in, systemic circulation levels. So there's just so much more to be done as we think about how we can really harness FC biology in the context of vaccines and therapeutics. Okay, and so with that, I can't thank everyone. I hope I showed a picture of the people who did the work. This was a village, and I thank you for your attention, and I'm excited for the questions.
Thanks, Galit. That was an awesome talk. I think there are a couple questions coming through, but as I mentioned, we'll wait till the end and have a discussion around there. And maybe Gestur, I'll pass it over to you. You should be able to
read the
share your
questions?
slides.
Should I read are we going to do the questions at the end, Lisa?
We are going to do the questions at the end, just so we have sort of a holistic discussion. There is a question that's come through so far. Go ahead, Gestur.
Thanks. So you can see my screen, and you can hear me well. Okay, thanks.
Looks beautiful.
Let's hope it stays that way. So thank you very much for organizing this fantastic lecture series and invite me to actually participate in the discussion. So I'm going to talk about IgG allotypes today and the polymorphisms in antibodies, and I'm very happy that Galit mentioned all the beginning all the swarms of antibodies, but now I can then skip the details and go into the nitty-gritty, perhaps nerd-like details into something that is really not our most active field of research, but we're definitely active, and so I'm happy to share this. So basically, as Galit mentioned, we tend to think of our antibody responses in terms of, at least in the IgG field, as mainly composed of 4 subclasses. They interact, of course, to complement, but in this case, I will mostly talk about Fc receptors, and there is this very complicated interaction phase. Because you have different types of antibodies, you have different post-translational modifications in terms of glycosylation, and the subclasses, of course, different Fc receptor on different cells in different formats and configurations. But this is very much complicated by the fact that the Fc receptor, for example, which is going to be a talk in the 2 weeks by Bob Kimberly, who is going to talk much more about the other side of the part. But simply I have to mention this because it's really, all this talk is also in the context of this variation, which is the variation in Fc receptor that are functionally differentiating how they actually interact with the subclasses. For example, this Fc gamma receptor 2A polymorphisms are very well known to actually depend and decide basically whether or not the IgG2 subclass is very good, capable of interacting with these receptors. And also allotypic variation of Fc gamma receptor 3 are very well known to have either high or low affinity interactions with IgG1 and IgG3. But in the last 10 years or 15 years, we've come to know and learn that it's much more complicated than that, because this whole locus on chromosome 1 of the Fc gamma receptors is in a continuous flux in genetic duplications and deletions, leading to this copy number variations, where you have basically this mostly 2 or 4 different or 3 main duplication and deletion areas, where you can have actually 0, 1, or 2, or 3, or 4 different copies of each of those variations. So basically, you can have an antibody profile or Fc gamma receptor profile, because this is with a double amount of Fc gamma receptor 3 or 3 times the amount, or even none. And you can have the open reading frame of Fc gamma receptor 2C, which is basically Fc gamma receptor 2B on the outside, but on the inside it's like an Fc gamma receptor 2A. So an activating one. You also have polymorphisms of the inhibitory receptor on B cells that are influencing how we respond basically to our immune response, and certainly in the secondary responses, so that it's affecting the expression of those receptors, and whether or not they're in lipid drafts and the signaling cascades of thereof. And Fc gamma receptor C also seems to be able to be expressed on those B cells if an individual actually expresses this variant, which can basically have an opposite effect of the inhibitory receptor, because it basically stimulates the response. And also there are individuals actually that have a negative sort of a Fc gamma receptor 2B, which is non-active, which we recently described here. But that's very uncommon. But that's not what I'm going to talk about. I'm going to talk about the allotypic variation on the other side, on the IgG side, and which is on the protein side, but also on a slightly end up on the last bit, which is the post-translational bit, basically all adding to the variations on the antibody sites and how they interact with the Fc receptors. So basically, as you know, so the 4 subclasses, they have a very well-known set of mutations that we know for a long time. Actually, before we even knew that there were polymorphisms in the Fc receptors, actually some of those variants were in the 80s and even before that used in paternity sort of a discrimination and even crime scene investigation before we knew the HLA variation and we could sequence DNA very well. But that has long been forgotten and nobody really cared about looking into what does this actually functionally mean. So we can see that the variations of those across the different subclasses is very much variable, but very strikingly that IgG3 is basically littered with these variants. And basically one of the first things that I would like to draw your attention to are the main differences in IgG3 compared to the other subclasses, which is the short half-life, a slow or low placental transfer, something that we looked into more than a decade ago, actually. But then just a short introduction to the neonatal Fc receptor, which is very important for its half-life of antibodies. So it's an IgG receptor on the inside of cells, and actually even if the FcRn would be on the outside, it wouldn't bind because it requires an uptake of the antibodies and a fusion with those vacuoles with FcRn-containing vesicles, and it requires a pH of lower than 6.5 when you get protonation of histidine residues, allowing for IgG binding, which then triggers FcRn to actually transport the antibodies back to the outside, where it's released again at pH 7.4. And basically the net effect being recycling or transcytosis, depending on the cell type. So basically, it's important to realize that it's really pH-dependent pathway and the heart of this interaction. So with the Fc portion here, the neonatal Fc receptors over here, and for the remainder of the talk, Fc gamma receptors over here. But for FcRn, this heart of interaction is the histidine 435, which is protonated at 6.5 and become positively charged, and only then the neonatal can bind to this. So basically, you can also see that in all mammals that I think we know, we have histidine at this position because it's simply very important to keep the half-life of those antibodies, except for human IgG3, which has an arginine in most of these variants that we know of, except for a couple of them that are actually quite uncommon in Europe, at least, and some parts of the world is more common. And basically what we did is to look if this is something explaining why IgG3 has a short half-life. And basically, yes, it is, because when we looked at in transport of antibodies across the placenta or the cellular transport system, we saw that actually IgG3, when transported alone, was very sufficient in doing so, but only if you mix them with IgG1 and IgG3, you see that IgG1 is inhibiting IgG3 transport. But if these IgG1 and IgG3 have the same amino acid at this position 435, either an arginine or histidine, both are transported quite efficiently. If you have an IgG1 that cannot be transported because you make an alanine variant at position 435, which cannot bind to FcRn effectively, IgG3 is transported quite well. And if you reverse the whole situation, basically you see that this is sufficient to explain why IgG3 has a very short transport. Or low transport, basically. Now it's IgG3 that is inhibiting IgG1 transport. And actually in humans, there are agammaglobulinemic patients receiving IVIG therapy. We see that at the replenishing dose after 4 weeks, they receive it around every 4 weeks. You can see that the relative amount of IgG1, 2, and 4, at least IgG1, 2, the most main subclasses found in humans, is more or less in equal proportions. IgG3 is the bulk is basically gone because it has a short half-life, except for this variant that contains histidine, because the IVIG given is a pool from thousands of individuals is also present. And if you look at this specifically, you see that actually it at least as well maintained an IgG1 and IgG2, and also transport across the placenta of this allotype, not IgG3, or curiously enough, IgG2, which is also well known to be transported at low amount across the placenta. But at least the IgG3 of this allotype with an histidine is transported across the placenta. And so basically both the half-life and transport across the placenta is explained by this polymorphism, basically showing that what we classically have in textbooks is not completely true, depends on the allotype. So for the rest of the talk, I will talk, or most of the rest of the talk, I will talk about the other polymorphisms and basically try to, some effort that we did to Steven de Tije and people at GenMap to actually look at what the other polymorphisms do, if anything, in terms of Fc gamma receptor binding. And what we did in the first place was to use a Fc gamma receptor array where we flow the different IgG allotypes over this and just look at what it means for the different Fc gamma receptors. And basically for Fc gamma receptor 1, didn't really mean anything. So we see that for Ig1 and Ig3, they bind more or less very well, and Ig4 slightly less. This is something we knew, and IgG2 does not at all bind to Fc gamma receptor 1, at least in this monomeric situation. But when looking at the low affinity Fc receptor, we start to see something, but not really something remarkable, perhaps, for Fc gamma receptor 2a, the two different polymorphic variants. But once we looked at Fc gamma receptor 3a, then we see some variants that seem to have lower affinity binding. But strikingly, when you actually started doing ADCC experiments, so these are anti-rhesus antibody that recognize red blood cells, and we do ADCC by NK cells, we see that now the picture is completely different and not basically matching completely, at least with our affinity profiles. So we were quite curious about this and made a different set of all these antibodies against the 3-nitrophenyl antibodies, and basically haptinated our red cells, used the antibodies again, see basically more or less the same picture. There are some antibody variants that really stick out or actually have less effective functions than we would expect it. And basically, some of those things actually do correlate. We see some variants that have both lower affinity and have lower activity. So basically looking into this table, which these variants are, and these are those variants, and they are really sticking out because they have really unique mutations that possibly would explain this. And basically we went in and just mutated those IgG3 variants back. So we put in a tryptophan in arginine containing variants, and they actually lose their activities. And so in any case, even in IgG1, you also lose activities if you put in tryptophan into IgG1. So basically explaining this, and the same thing for this tyrosine variants over here. I'm not going into that in details, but I think what it's even more striking is this difference, where we basically see nothing happening in SPR or in affinity, but we do see a striking difference in ADCC activities, either going up and either going or down. So basically, the ones that are really sticking out in terms of increased activity in ADCC is this variant that lacks basically 2 exons of the hinge. So in Ig3, you can normally you have one exon encoding for a hinge in all the subclasses except Ig3, which normally has up to 4 exons, but this variant only contains 2, so it's very short. And the other ones that actually has a lower activity is this variant that is very unique in having leucines at this position 291. So basically, first this variant with the short hinge. Basically, if we make a different set of antibody, like anti-CD20 antibodies with either 1, 2, or different types of a number of exons, you see that the shorter the exons are, even down to an Ig1, the better the ADCC seem to be, which basically reminded me much about a recent publication from Mark Craig's lab in Southampton, basically including CD20 epitope at different lengths from the membrane, and basically seeing that the longer you increase this distance, you actually lose your ADCC potential, but you increase phagocytosis. So basically, perhaps something similar is happening here with an Ig3 with a long hinge. So if you elongate this distance by elongating the hinge, you may actually lose the ADCC potential. And actually, in HIV-infected target cells, recently been published, is that the longer the hinge, basically you get better phagocytosis, very similar to what Mark Craig was describing, but also lower ADCC if you elongate your hinge. And in COVID-19, there have been some reports of having the shorter hinges of Ig3 is also associated with severe disease and ICU deaths, or when discriminating between survivors and death. And in cerebral malaria, a complication in malaria, you basically also seem to see that it's associated with pathology, the shorter the hinge. Although we don't understand completely what's happening here, at least it's striking that it has a very big difference and even surpassing IgG1 antibodies in here. But these antibodies here with a lower expected ADCC potential, while seeing no difference at all in affinities, puzzled us. And we went here for an assay where we looked also at avidity, simply asking the question, are we simply missing something using only one single binding molecule, on a one-to-one basis, instead of we went over this chip with the whole cells, opsonate cells, and looking at the binding strengths of these whole cells to this chip on the different flow rates, and basically plotting this as area under the curve. And basically what we see is that if you put in those variants that contains leucines at this position, all of these antibodies lose this avidity, while with the control anti-kappa antibodies, you basically see they're opsonized the same way, but they simply lose this avidity effect, both for Fc gamma receptor 3s, both polymorphic variants, and Fc gamma receptor 2. So basically, to sum up this up, I think it's very peculiar. Of all those variants that we see in nature, basically, is that they don't seem to do so much in functional terms, at least in Fc gamma receptors. And these variants that do something, they're all located at a position somewhere farther away from where the Fc gamma receptor are binding. And how they are actually doing this is something that is puzzling us, basically. But still, it's also interesting to note that all these variants, they are not distributed evenly across the globe. Some of those variants with increased half-life are mostly found in either Africa or Northeast Asia, and some indigenous in America, and other variants with, they all have different sort of locations across the globe. But then the question is, what does it mean in terms of pathology or protection in infectious diseases, is something that I think future studies will more look at, and I hope at least the length of the hinge of the IgG3 seem to really matter in here. Then to the question, how can those variants affect effector functions without actually being in the interaction place for Fc gamma receptor? Perhaps one of the reasons are allosteric configurations of this DE loop that is really a very critical loop in interacting with Fc gamma receptors, but also it harbors the ending glycan that is also very important. And perhaps it's also changing the accessibility of glycan modifying enzymes that actually also affect the antibodies. And I will go through some of those things in the last few slides. And basically, Galit mentioned this also a moment ago, is that the glycan code basically is also very important. It's not only the protein phase of the antibody that is affecting the effector function. In particular, core fucosylation is extremely important for binding to Fc gamma receptor 3. You basically, if you engineer antibodies with a different, with either fucose or no fucose, you can see that all the other glycan adducts that can be changed, so basically increasing the number of galactoses, the bisecting glucanac, or increasing the amount of sialic acid that is not basically shown here, it doesn't mean anything except if the antibodies is afucosylated. And basically then you see a huge bump in affinity to Fc gamma receptor 3, and it's not happening to other receptors, and this is because Fc gamma receptor 3 is unique in having its own glycan that basically needs to be accommodated here into the structure, and where with the fucose is basically in the way. And only if you take off this fucose, you basically accommodate for this glycan, which is very curious because almost all of our antibodies in our blood contains this fucose. So this is a very unique type of antibodies, but it's definitely found. And what is important is to note is that once you make such an antibody response without this core fucose, so these antibodies are much more active, at least in terms of ADCC potential of NK cells. So these are, for example, engineered antibodies completely identical on the protein backbone, either engineered with fucose or without fucose, and basically no matter what concentrations you throw at this, only the afucosylated antibodies mediated ADCC. And this is a type of immune response basically that we've been seeing in all kinds of alloimmune responses in pregnancy, so in anti-rhesus, causing anemia, life-threatening anemias or bleeding when they target the platelets. But they also, so you don't have to be really pregnant to make this, because we also see this after blood transfusion, incompatible blood transfusion of either red cells or platelets. You make afucosylated antibodies against HLA also after such a transfusion. And recently, Marge Ackerman also showed that in transplantation, you also make this afucosylated response. And once you do that, that is also a critical, seems to be a critical factor in determining basically rejection of those grafts. And what we've been seeing is that you have basically two types of immune responses, either the ones that are fucosylated, and those are the bulk of your antibody response. 96% of your antibodies are fucosylated. This is the bulk. These are the antibodies against the proteins, soluble proteins, inactivated viruses, meningococcal proteins, polysaccharides, autoantibodies, etc. They're all fucosylated antibodies. And only the categories basically that we've seen, and actually something that triggered us is an old study from Marge Ackerman and Galit, where they looked at anti-HIV antibodies in elite survivors, where they showed that you also have this type of afucosylated responses. And later, actually, in dengue, by Jeff Ravitch and his group with Taiwan, showed that you actually also have this afucosylated responses, which basically led us to the hypothesis that perhaps the similarity between these allo responses and the envelope viruses, and not the non-envelope viruses, is this presence of foreign antigen in the membrane of self, and perhaps that something of a self is required as a co-signal. So basically, we showed this also, in all kinds of all the envelope viruses we've seen so far, CMV, HIV, RSV, mumps, measles, HPV, and you name it, also SARS-CoV-2. There seems to be a transient response in SARS-CoV-2 because only mostly in ICU patients, you see a transient response. When they seroconvert, they have a huge amount of antibodies. They very quickly, and they are afucosylated that quickly, after a few minutes, actually become fucosylated. And in this time, that's a very critical moment where they have a huge amount of antibodies, huge amount of afucosylated antibodies that are highly active and seems to cause a pro-inflammatory phenotype. But also the same thing in dengue fever, as a group of Jeffrey Ravitch has shown. Which brings me to the question that was addressed here a few weeks ago by Arturo Casadoval on plasma therapy, and what Galit now just mentioned is that plasma therapy in such a disease, such as SARS-CoV-2, you not only have to think about the quantity of the antibodies there, but also the quality. And knowing that these responses can also be partially afucosylated and associated with severe disease, although I think that they can actually also be protective, but under the circumstances when you are severely ill with a proliferating virus, it may not be such a good idea unless you have an antibody that you give that is highly active very early on in the infection, that you can actually give protection. But just to realize and get some of this alternative picture that also Galit been showing, that the antibodies come in different flavors. And this is one dimension that you see in this convalescent plasmas that have been given to patients, that most of the people that have been hospitalized, they make huge amount of antibodies, so on the x-axis, but they're also very differently varied in terms of the afucosylation level, which also determines their effector functions through Fc gamma receptor 3. Also, I think it's very important to note that the strange feature of having those antibodies in pregnancy, which basically in nature should not be tolerated from an evolutionary perspective, if I may, but perhaps only been tolerated if it's important for diseases such as envelope viruses, as we see these responses, but also for malaria, because these alloantigens really resemble those antigens in malaria cells that actually are infected and expressing those knobs, which encodes proteins that are very important for nestling of those cells, infected cells on the endothelial cells. And basically where we see the amount of afucosylation is staggering, never seen before, down to 10%, actually very similar in autoimmune diseases in pregnancy. But now it's not from 100 down to 10, but it's everybody is making this kind of afucosylated response, and which seems to be correlated with protection in malaria in endemic region. But once you take the same antigens and actually immunize humans, you don't make this afucosylated response, you make fully fucosylated response, showing that the context basically of these immunizations, the natural immunization on red cells seems to be important, as we hypothesized that. So basically, if you want to make a good immune response against malaria, you should not give them as a soluble protein. And then the question is also, what about COVID-19 vaccines? Because basically what we're doing here with those adenoviral vaccines and mRNA vaccines, we're giving them to host cells, basically producing those proteins and giving them in the exact same context that we have been hypothesizing that actually it's required to make afucosylated responses. And actually this seems to be happening. So I'm not going into the amount of antibodies, this is well known, but basically we see in naive individuals in blue, all of them make afucosylated antibodies after seroconversion that quickly goes up, which is very much similar to what we see in hospitalized individuals that actually do the same thing. So they start with afucosylated responses, and then they go up basically in weeks' time, which is less so for people that actually have mild infection. Curiously enough, that people that actually are SARS-CoV-2 experienced, so they actually had an infection, then got vaccinated. So this is with the BioNTech vaccine. They actually start with high level of fucosylation that goes down. They've increased this number because you see the magnitude is perhaps not striking, but it's definitely there. They go down. But the naive individual, they all start with these vaccinations with low core fucosylation. So we know have some handle to make afucosylated responses. We simply don't know how to retain them through vaccination. The last bit is on galactosylation of antibodies, another flavor of antibodies, but not through fucose, but through galactose, which is very normally around 50%. If you get infected by SARS-CoV-2, you make extremely high level of galactosylation, once you seroconvert, also through these vaccinations. And basically, only if you have our ICU patient, you drop very quickly, which is extremely curious. And the question is, of course, what it means. We and others have been showing that basically galactosylation seems to be a principal component in the antibody repertoire and determining the amount of C1q binding and all the downstreams of complement cascade, the deposition of C3, C4, and the lytic pathway of complement. So it doesn't matter how much fucosylation you have, the bisecting GLUT9 doesn't matter, but only the amount of galactose seems to be really determining this, perhaps with the slight addition of sialic acid. And so basically to understand what is happening here, why is galactose doing this? And it's important to realize that how antibodies activate complement is on surface of cells mostly. They suck each other up through the Fc tails, forming hexamers, and basically bringing the questions down to how are these antibodies doing this? Galactosylation doing this? Is it enhancing the inherent binding of C1q to those the globular heads, or is it changing the structure of the antibodies so the C1q can interact better? Or is it interacting, allowing the antibodies to better to form these hexamers and therefore activating and binding to C1q? And because of time, I'm not going into the details, but basically we have recently shown, and also a group of Genentech showed that it's actually the latter. It's basically the galactose here in yellow, two Fc fragments, basically these galactoses. The more you have, it basically seems to stimulate these antibodies to seek each other up through the Fc interaction, form hexamers, bind C1q, activate C1q, and do the whole kabam downstream of that, which is basically marking the target cells for downstreams activation. And then just to end here with coming back to the allotypes, what it has to do with allotypes? Basically very little. If you think of in terms of all the different allotypes and we produce them in hex cells, we see no change in fucosylation. All the antibodies are fucosylated normally if you produce them like this. There is some variation by section, but Galit isolation is actually very interestingly very much dependent on the allotype, and so it's isolation. And you can see that basically if you make them antibodies as anti-rhesus antibodies or TMP antibodies, they very have similar actually Galit isolation features that may actually be an important feature in how the antibodies are different from people to people, because you know that Galit isolation is going down with age, which is associated with then probably with lower effectivity of these antibodies to actually stimulate, complement, and protect against infectious diseases, because we know complement is very important for that, especially for encapsulated bacteria. But also here, just to end with the fun fact that these polymorphic residues that we found in this DE loop also affecting ADCC potential and Fc gamma receptor binding are also these variants that of these allotypes that have enriched Galit isolation. So perhaps this is the reason. So to sum up, antibodies are very important facilitator of target recognition. The IgG polymorphism can affect IgG half-life, so that's in the IgG3 allotype, and the polymorphism in IgG affect the binding and avidity of Fc receptor of effector functions through the Fc receptors. So it's basically this interface of Fc receptor and IgG is in a polymorphic state, and so each individual can be very much have different makeup of the Fc receptor, but also the antibodies. This is important to realize, and that the quality of those antibodies is basically also largely controlled through the glycosylation, which is really dependent on how the antigens are presented to our immune system. And this is basically especially fucosylation, which is really important, and that's through Fc gamma receptor 3. And we need to understand, I think that's one of how to understand how to induce this on purpose or prevent this on purpose, because in this way you can either stimulate pro-inflammatory antibodies when required, for example HIV, malaria, or actually to prevent this to actually happening where you want an antibody response that is more quiescent. But also it's not only Fc receptor, it's also complement that is important to think about here. So quality and quantity do not always go hand in hand. And I think that's a very nice match and echoing what Galit was saying. And with that, I think I would like to thank my PhD students and postdocs, technicians throughout the years, and collaborators throughout the world, but mostly Manfred Wührer, who has been my eyes and ears on the mass spec, and Theo Rispens also for the allotypic work. And our collaborators at GEMAP for the polymorphic variants. And with that, I thank you very much for your attention.
Really interesting talk, Gestur. That's awesome. Although sugars are always just so complicated. All right, so I think we've got some time for questions. We have 15 minutes left in our time. And Galit, you answered a question. I don't know if you want to expand on that or add anything to it. I'm assuming everybody can see the Q&A.
Yeah, I think it just gets to the point that in that acute infection model, we didn't see huge subclass differences in kinetics across survivors and non-survivors, but we did see as differences in like accelerated induction of FcR binding IgGs. So I'm assuming it has to do with Gestur's observation of some sort of glycosylation change that occurred preferentially or faster in survivors than non-survivors, and that those may have given those individuals advantage in recruiting particular Fc receptors to mediate opsonophagocytic phagocytic clearance of the virus. So that I think that's the assumption. We didn't at the time. We didn't have enough material to do glycosylation profiles, but then I knew Gestur and Manfred would come to the rescue, and they did.
So maybe I'll ask a really provoking question to you both. So, Galit, you had started with sort of this 140 number, so the 4 subclasses, and then the 36 different sort of bits of sugars that could be combined on all of those. My question is, are there really 140? Or is it more nuanced in that, take one subclass could you then have just a subset of those sugars that would sort of appear versus the whole 36 bits? And, that might vary over time, like the age data that you showed, Gestur, but I just wondered what you both thought about that and whether or not it might be a little bit more narrow of a definition. We just don't have it yet.
Do you want to go first? Yeah.
Oh, okay. So is it 126? I would say if you really go to the extremes and really calculate on both sides, because you have symmetry, both, of course, 30 something, if you only talk about one side, but if you talk about two sides, you actually amount to 666, I think, exactly.
So that's an interesting number.
But I think exactly, but I think that's not really a realistic number in terms of because the whole machinery that is actually putting these on and taking the things off is the same B cell on both sides. So I think that the antibodies are likely to be more similar on both sides. And I think also in terms of functionality, I think everything is pointing to those at least two major sugars, or perhaps three, if also salicylic acid is modulating this. But fucose and the presence of the fucose or the galactose is really, I think, number 1 and 2, basically. So it's there are more variations, but the functionality, I think those are the top two. But I think we're always in for some surprises down the road. Who knows?
And I think to get to this, I mean, so Lisa, we don't have technologies yet that give us for a single antibody, for a single chain, right? What is the actual structure? Mm-hmm. And so I think right now we're still looking at aggregate data, always in all this data. And so, when you look at a spectrum from a stochastic sampling of a particular blood vial, right? What we're seeing is that there are clearly some structures that are more abundant. There's no question about that. And there's I would say about 19 to 21 structures that we see like at very high levels in most individuals, and then they kind of fluctuate, right? How much salicylic acid, how much fucose, right? How much galactose. But I don't think that we're really at a level where we can say are all the structures really present? And if they are, would they be on both arms? Would they be on one arm, right? So I think there's still some tech dev that is required. And to Gestur's point, I think it's really important, right? So we are now skimming the surface. We see we know the variation theoretically should exist, right? And the variation is huge, right? I'm probably grossly underestimating how many combinations we possibly can have. And that's only on IgG. Right? Because now we're learning that IgG glycosylation obviously makes a big difference, and they can have up to 100 structures per arm, right? That's even crazier. And then you have O's and N's, right? And then IgM, we're seeing that there's, variation in IgE, or Rob Anthony has shown us clearly there's some sites that are driving anaphylaxis. So I think that, I think the point is, I think that, given where we are now with the technology, I think we're starting to learn that there is this kind of functional impact of these changes, right? Which is what Gestur's talking about. And we're starting to see that this is controlled, genetically it's controlled by age, it's controlled by vaccination, it's controlled by all these different kinds of, processes. So it's all within the realm of potential therapeutic, to leverage this. But the question is how much further can we push it if we had technologies that allow us to really understand how this is leveraged naturally during immune responses. So I feel we're just kind of skimming the surface, but the data is super cool.
Yeah, and I was really glad, Gestur, that you brought up, how do we design vaccines to induce the right type of immune response when it comes to antibodies in particular? It's just a fascinating question overall. And that's in the context of a pathogen that you might get infected with a lot versus one that, you might only encounter once or not at all. And that could be really interesting. So whether you come in there as a naive individual versus somebody that's been exposed over time is also super interesting.
Exactly. I think what we don't know here is also the timing between the initial immunization event and then the second one. So I think early on in the COVID-19 pandemic, when we started vaccinating, many of these people that actually saw the virus had recently got the infection. We don't know, would this all be completely different if we would have waited with the vaccination in these individuals? We don't know.
Yeah, it's really interesting. I'm always struck by those, all those studies we've all done where we, take sets of mice, and you give them a bit of vaccine, say it's flu, like you give them their flu vaccine, you infect them and they get a little infected, or you really almost like they're, almost to the brink of dying, and just comparing the immune responses across those three groups, just really interesting. There's another question that came through. So this is for Galit. You mentioned IgA, hot topic right now, given some of the papers that have come out recently on nasal vaccines, especially. But IgA and its receptor expressed by newts are important after immunization. And since IgA deficiency is very common among the population in Western countries, do we know whether IgA deficiency compromises the efficacy of the mRNA vaccine?
So the answer is, I think people have looked at this pretty carefully in immune compromised populations. There's no evidence that individuals that have an IgA deficiency really show any clear evidence of more severe disease. And I think the reason for that is that there's just redundancy built into the system. So that's not to say that IgA is not essential and that IgA cannot be a critical player, particularly against mucosal pathogens. It just means that these individuals have, backup mechanisms that have been evolutionarily programmed in us to essentially make sure that we don't, die if we come in contact with a mucosal pathogen. I think what's going to be very interesting, though, is to see, right, in those kinds of individuals, do they just have, more transmission events, right? Maybe they don't, have severe disease consequences because they got great T cells and they've got great IgGs and anamnestic B cell responses, but maybe they are more likely to, get reinfected, right, over and over again, because they don't have this potential barrier at the respiratory, the respiratory tract. But I think that the data, at least from severity of disease and death perspective, it doesn't seem that IgA is a complete wipeout of protection. So I think that's encouraging. And I think it's good, right? We have lots of G's that go to the nose, right? We make lots of G's all the time, and we also make M's, and M's also work extremely well mucosally. So I think that there are these other ways that we're finding to fight these pathogens. Joe, you have a question.
I do. So first, I also want to say thank you both for your presentations. They were fantastic on an incredibly difficult topics. My question is I guess it's mostly for Galit, but I'd like to hear Gestur's thought as well. Is you had described, early on in the pandemic, you had done some experiments with people who were either who had gone to ICU or survived, right? I'm sorry, survived or didn't. And you pointed out, phagocytosis as something that was seen in your survivor population to a greater extent than in the non-survivor. The question I have is, all right, so it was a pretty stark correlation. So it's really a twofold question. One of them is, have you been able to follow that up into people who had not gone into the hospital, so never got to the point where they were so sick that they were in the hospital? And is that a correlation or is actually the cause of survival, or what allowed them to survive? Or was it just that the people who didn't survive couldn't do that, or they had already tried that and they had moved on to something else because that didn't work, right?
Exactly.
That's a great question. So let me answer quickly, because Gestur has some data also about this. But what we did, Joe, is we moved as the pandemic moved on to other populations. So we started off with severe, and then we went to non-hospitalized, and then we went to community. And what was amazing is that as we moved in these different populations, what made the least sick people unique is they all had enhanced binding to an Fc receptor involved in phagocytosis, Fc gamma R2A. It was super selective, and it was always on their S2 antibodies. It was crazy. So in the mildest cases, people were completely asymptomatic in our community surveillance. It was wild. They had these really high S2 specific antibodies that had this enhanced Fc gamma R2A binding opsonophagocytic function that we think was derived from cross coronavirus previous immunity that allowed them to essentially respond to the virus immediately and just get rid of it immediately from the system. They barely even made a new response to SARS-CoV-2. That's what even like was completely like baffling to me. They were basically just using this pre-existing immunity to S2 to get rid of the virus. So that was really kind of profoundly like illuminating that it was, you know, that these really sick people are leveraging this specifically for SARS-CoV-2, but the really mild individuals already have this baked in because they've seen OC43 and other viruses in the past. To answer the question, I think that Davide probably did this experiment best with Vir, where he did with the folks, he basically took antibodies, I think, to S2 and gave them Fc receptor binding capabilities and showed very clearly that when they had these Fc receptor binding capabilities, that was super protective in hamsters. And so he was able to show there that these types of opsonophagocytic antibodies really can causally control and clear the virus more effectively in a pathological model of the viral infection. So I think that, and there have been repeated studies over and over again. We did a couple, but I think that was the best study that I saw, which was a beautiful paper that was published with David and Davide. And I think that that really was kind of the proof that FC matters, especially for particular targets that might be more VOC specific. Gestur, do you have any thoughts on that?
I thought what you just described is really spot on. I think that's very important. This pre-existing immunity that is very likely something that is allowing people to kick in earlier, to actually respond quicker. So it's probably what we're measuring in this terms of kind of a rather a-specific antibodies. They're not really that functional, but it's probably for some part good enough, but also a reflection of underlying T-cell immunity that may also help to kick in, to kickstart the new antibody wave that are really, really specific and cause this, that the initial wave of the proliferation of the virus is not going completely uncontrolled early on. I think that's really what is happening, of course, in the ICU cases, the really, really severe individuals. But there we see two things that are really unique, I think, and that's the massive amount of antibodies that they produce, and also the type of antibodies, which are indeed highly more afucosylated responses than you see in individuals that don't go into hospitals. They do never make such low levels of afucosylation as we see in the people in the ICU. There is not huge difference, I must admit, but there is a difference, and I think the amount of functional difference between those afucosylated responses and fucosylated ones is sometimes so staggering. It's really coming from 0 to everything, and we've seen this a couple of times, both in tumor models, in vitro, but also in vivo models, with the malaria antibodies looking at degranulation of NK cells, and in our alloimmune models and destroying red cells. Some of these antibodies, actually most of these antibodies, they are completely devoid of function unless you take off the fucose. So I think that's staggering. So I think even though it's a low amount of afucosylation, so down to 70% of or 80% of fucosylation, it can be functionally quite significant.
Awesome. And with that, I think we're at time. So I'd like to really extend lots of thanks to Galit and Gestur for really innovative talks and for not sugarcoating everything you're talking about. Just kidding, had to do it. But thank you everyone for